So Travis was an ARCS scholar in 2009 to 2010 at Scripps Research. And I love to put that in context with ARCS. At the time, Candace Cole was our president. And Travis, you may remember Candace, she's still an active member. She's not in attendance today, but you may get to see her again. Travis earned a BS in biochemistry from Boston College and a PhD in chemical biology at Scripps Research, where he was an ARC scholar. He was at Harvard Medical School on an NIH fellowship in the Department of Biological Chemistry and Molecular Pharmacology with Dr. Christopher Walsh. Then in 2012, Dr. Young returned to San Diego and joined Calibr, which stands for the California Institute for Biomedical Research, which is part of Scripps Research, as a principal investigator. And today, almost 10 years later, he's the vice president of Biologics at Calibr. He also holds an appointment as professor of chemical biology in the Department of Chemistry at Scripps Research. And Calibr is a first of its kind nonprofit translational research institute that is dedicated to creating the next generation of medicines. And Travis will tell us exactly what that means. In fact, the title of his talk is very aptly named. So Bench to Bedside Immunotherapy. Over to you, Dr. Young.
Great, thank you. I appreciate that introduction. Yeah, I'll talk somewhat about what Calibr does. Can everyone see that? Is everyone seeing a full screen PowerPoint presentation? Yes. Great, excellent. So I did my PhD at Scripps. And I think everyone here is familiar, obviously, with Scripps, but maybe not familiar with everything that Scripps has done or the things that have come out of Scripps. So there's been a lot of great innovative work that everyone's familiar with, but out of that have come a number of FDA-approved drugs. 10 drugs actually have come from work that has been developed, or the science or the underlying mechanism for the science has been developed at Scripps. And many more that are in clinical development with various pharma partners. So I think what we had looked at over the years of Scripps is this incredible innovation, but Scripps was not putting molecules themselves into the clinic. In order to do that, most of the time Scripps would out-license to a pharma partner or spin out a biotech that would be backed by venture capitalism. And so, you know, the challenge was clearly, how do we take the interesting science that's being done at Scripps, and how do we ourselves take these homegrown programs, and how do we put them into patients and demonstrate their worth? And in doing so, realize some of the potential equity from those programs, bring it back to the nonprofit, bring it back to Scripps, and continue to fund and fuel innovative new work here. Rather than seeing our programs be successful, but be successful in others' hands and perhaps not capture the full value of those programs to be able to bring it back to the basic research that we do here. So that's how we developed Calibr, and we developed it not just on one technology that came out of Scripps, like would be done with a typical biotech, but rather we focused on a huge wide range of different diseases, including degenerative diseases. Osteoarthritis was one of our first clinical trials. We have got work going on in childhood diseases, autoimmune diseases. We had a big relationship with the Juvenile Diabetes Research Foundation, the JDRF. Infectious diseases, we have a big relationship with the Gates Foundation that I'll talk about. And then cancer, where my group is mostly focused on the development of new immunotherapies, which I'll talk about today. And so Calibr became this model to bring all of these different fundamental areas of science together under one roof, and to provide a conduit to get from the bench to the bedside to allow patients to potentially benefit from these novel therapies. We've now done this successfully. We've got these different disease areas that we're working on that are listed there in the middle. And we've got Calibr, and in the way that I've described, we somewhat serve as a bridge between the innovation that goes on at Scripps and the early stage clinical development that can potentially bring patient benefit. And so that's where we focus. We focus in interactions with the FDA, in developing investigational new drug applications. We focus in the manufacturing of the drug products. We focus in more what comes after the basic science in order to get to a patient benefit. We've built a lot of different capabilities. If you think about a small biotech, which may have 100, 125 people, and it focuses on just one thing, maybe one technology, one indication, one disease, and we're focusing on a huge range of different things. We have just about the same number of people. What we've had to do here is build all of these different pieces of the model in order to have the infrastructure to do this. And the idea behind this really came from Pete. This is Pete's brainchild, Pete Schultz. And it came a lot of this from his experience in running GNF, which is the Novartis Institute, which is across the street from Scripps, which many of you are likely familiar with. So there was a lot of learning that Pete had over the decade from around 2000 to 2010 that we were able to bring into Calibr when we started Calibr. In sometime around 2012. So we're almost 10 years old now. If you're familiar with the different modalities for drugs, there's protein-based drugs, antibodies like Humira. There's small molecule-based drugs, which is basically anything that you take as a pill. There's also gene and cellular therapies, which I'll talk about today. On the left-hand side, on the discovery side here, we have programs in each one of these areas. And then in the optimization side, this is the central component that I talked about, where we bring things to the FDA, we discuss it with the FDA, and then we eventually bring it to the right-hand side here, which is bringing it into our phase 1 clinical studies. So these are the different infrastructures, the different core areas that we've developed here at Calibr, again, with the goal of bringing things from bench to bedside. I'll skip over that slide just in the interest of time. So someone yesterday, they said, well, there was a press release recently. I forgot whether it was maybe Holly or Ingrid. Someone said there's a press release yesterday on the amazing work that Calibr is doing in COVID research. And I said, I wasn't actually familiar with that. And I think it goes to speak about the reach, the breadth that Calibr has had in doing all of these different pieces of diverse work in the nonprofit world. And I think it's really unique that we built this model, which is like a biotech in the nonprofit world, and linked it up with Scripps. And this is one really good example of it, what led to this press release. One of the great relationships we have is with the Gates Foundation. And they have funded us to work on several different third world indications. And then when COVID hit, they funded us to do a lot of COVID work. As part of that, I think everyone's familiar with the cost and the time and the complexity it is to develop a drug from idea through to an approved product. And so one of the ideas that came up working with the Gates Foundation is, what if we take all of the molecules, all of the drugs that have ever been dosed into a human, whether they be approved medicines or whether they be experimental medicines, whether they be done in the US, in Europe, or China. What if we took all of that information and we consolidated it into a library of drugs, which represents everything that's ever been in human? And Gates Foundation funded us to do the data mining effort around this, and then funded us to actually build this small molecule drug library, which is about 13 to 14,000 drug compounds. And the requirement was that we provide this free to any nonprofit researcher worldwide who is also agrees to then share the work publicly within the first year of them developing it. And so that's where this press release actually had come from with our COVID work, because our reach, as you can see down here with the graphic, and I think I've got a bigger piece of this graphic here, is really been worldwide. We've shipped this library out. This was really instrumental in the early days of COVID, especially in testing whether or not different approved medicines would be effective against COVID. And everyone's likely familiar with the various pieces in the news of that have been throughout the last 14 months or 15 months of, is this drug, for malaria, is this going to be appropriate for COVID? Is this going to be appropriate for COVID? Well, this was one way in which we did it And we were already set up to do this. I think that's the other really important thing is we were already set up to do this prior to COVID. We had been working on this for several years in advance in collaboration with the Gates Foundation. And so I think, again, it just goes to speak about the reach and the impact that Calibr and Scripps has had in basic research around the world. Today, I'll tell you about very briefly about two programs that have come from my group that are currently in clinical studies. And they exemplify somewhat how we've taken things from either internal research or from Scripps, and we've enabled them to get to patients. The first one I'll tell you about is a therapy called chimeric antigen receptor T cells, or CAR T cells for short. It was actually the first approved gene therapy in the United States. Novartis holds that title with a product called Kymriah, which was approved in 2017. And it was approved for patients with blood cancers, ALL or diffuse large B-cell leukemia. Another smaller company called Kite, which was eventually bought by Gilead, developed the next product in 2017. And since, we've seen a string of approvals. And they have had absolutely remarkable efficacy, these approved products. They are mostly used in patients who have failed every other prior line of therapy for blood cancers. And they can induce remission in those patients that have failed everything else in up to 90% of patients. So it is really, really remarkable efficacy. But there's challenges with these therapies. It's a genetically engineered therapy, and I'll show you exactly how that's done in a moment. And so in genetically engineering a patient's cells, what can happen is the cells, they're autonomous. They can overreact against the tumor and against the patient and cause some really significant challenges. And so when we looked at this, we said, how do we maintain the potency of the product while also controlling the safety of the product? And can we invent a technology that goes into this existing technology in order to both get that safety and efficacy? So this is how this new class of drugs works. It's really truly a living drug. It's a living medicine. Patient comes into the clinic. This is after they would have failed the prior approved medicines, and again, this has been approved in B-cell cancers and blood cancers is where it's most common. They undergo a process called leukapheresis, which is where their blood is taken, the white blood cells are isolated, and it's sent to a lab. And in that lab, that's where the genetic engineering happens. They use a lentiviral vector, which is actually the same virus, which is, it's basically a deconstructed HIV virus. And they use that to transfer genetic content into a white blood cell known as the T cell. And then they expand these cells, they grow these cells in the lab for a period of time, then they freeze them down and they ship them back to the patient. And what this enables those T cells to do is it enables the T cells to seek and destroy tumor cells in the body. And so the drug is no longer a pill or an injection. It's the patient's own T cells that have been genetically engineered to seek and destroy tumor cells in the body. And that's why it's so incredibly potent, is because these cells, they do what they're meant to do, which is to clear infections typically, but now they're clearing tumor cells. So they go around the body, they surveil the various cell types of the body, they find tumor cells, and they eliminate them. This is getting into some of the data, which again can be remarkably efficacious. If you look over here on the right-hand side, here's a patient's scan, and you can clearly see very, very large lymph nodes here that are PET active, indicating tumor in them. And just 30 days later, complete clearance of the tumor. In fact, the first time that they did this, there were anecdotes from the physicians where they did the scan at a month later, and they said, "Is this the same patient?" Because there's no more tumor that can be found. But in doing so, when the T cells activate, they're part of the immune system. They're part of the white blood cells, and so activation of the immune cells to clear all this tumor burden can have some really significant side effects. As you can imagine, overactivated immune system can cause different types of autoimmune, and it can cause flu-like symptoms that can actually be very dangerous. Interestingly, one of the first things that patients present with is they present with the inability to write. They have some types of aphasia, they can't speak. And so one of the tests to see if a patient is going to experience one of these adverse events is we just ask them to write a sentence. And if they can't write, as you can see here, it means that they may be experiencing an adverse event, and that adverse event may get worse over time. And over here, I'm showing in this blue line here, you can see the patient's temperature spikes really quite significantly to over 39, almost 39.5 degrees Celsius. Now, this can be treated. There are drugs that can treat this. And after the initial about 1 week period of time, the patient can recover. But unfortunately, this has caused some patient deaths because the T cells, they have a runaway activation. So what we've developed is this control switch. We've developed a switchable system which can turn on and off the T cells, the genetically engineered T cells specifically, almost like a remote control. So the patient would come in, they get leukapheresis, just like I showed you in that diagram. They get the cells manufactured in the lab, they're sent back to the patient, but now they don't turn on until we deliver something called an antibody-based switch molecule. And that turns the cells on and allows them to redirect and to kill the tumor. The advantage here is that if the T cells, they get overactive, they're not active without this blue switch molecule. And the blue switch molecule is dosed just like a conventional drug would be dosed via intravenous injection. Once we stop dosing that, the cells themselves, they're inactive, and so they turn off. And so if there is an overactivation of the CAR T cells, we simply stop giving the drug and they'll turn off. And then we can turn them back on again later in order to capture some of the effectiveness of the drug. So, you know, I'm really delighted to say we've worked on this program for about a decade. And this was one of the first programs we started when we opened the doors here at Calibr. And we've been successful at bringing this into patients, and we've treated now 3 patients and are on to our 4th patient that we'll be treating shortly. In totality, we expect to treat about 30 patients over the next 2 years. It's a phase 1 trial. We're working with multiple sites over the entire US, but UCSD is one of the sites here that we're working with and actually enrolled our first patient. So we're very thankful to have very good colleagues in the area. And so, like I said, this trial will go on for about 2 years. It's open to patients with blood cancers, and we're really excited to see how this program works. In the interest of time, I won't take you through again how this works because it's somewhat of a recap of what we went over before. But we're again really enthusiastic to see the results of this trial. We're running another trial in prostate cancer, which I don't have time to talk to you about today. Also open at UCSD. Also uses a patient's own immune system to tackle the tumor, and we're just about a quarter of the way through the first half of that trial. And if anyone's interested in learning more about that, I'd be happy to talk about that offline or after this. Happy to answer any questions. I know I went over a lot of material in that short period of time, but you know, happy to talk about either Scripps or Calibr or the clinical study that we're running now.
So anybody have a question? You can enter it in the chat box. I guess I'll pose a question, Travis, while people are thinking. Can you predict what might cause, which patients might have an overreactive immune system? Is it linked to age, for example, or do you have to basically wait and see and then adjust the dosage so that you can control that reaction?
That's a great question. That's really the $64 million question that the field is after. No one really can tell yet which patients are going to have adverse effects or side effects of this therapy. We do know that children are, this was first approved actually in pediatric patients, I should say. And we do know that pediatric patients are much more resistant to the side effects. The side effects are, and what's interesting is that people have probably heard about what the side effects are now due to COVID. The side effect is similar to the overactivation of the immune system that's observed in COVID. It's actually called cytokine release syndrome, and it's treated with the same drugs. And so when we opened this trial, we actually had a challenge in getting those drugs because they were all reserved for COVID patients. And just like COVID, we know that children are less affected by things like cytokine release syndrome. So somewhat of a loose correlation with age, but otherwise we're not really sure.
Yeah.
Travis, I have a question from the audience. Are any T-cell therapies in development currently for brain cancers, such as glioblastoma?
Yes, there are. There are CAR T cells that are in development of City of Hope, which are similar to the approved CAR T cell products that I talked about in the beginning, but they have a little bit of a difference in that they're not targeting B cell or blood cancers, they're targeting glioblastoma tumors. And then Baylor in Texas is also looking at CAR T cells for brain tumors.
Good. Another question. In the CAR T, excuse me a minute. I just lost my chat box, but I'll get it back. In the CAR T cell program, do these engineered cells remain in the body forever, either active or inactive?
Yeah, that's a good question. In some cases, they will remain in the patient for very long periods of time. And in fact, we follow patients for 15 years to see how long those cells will remain. In other patients, they don't remain very long. Luckily, there's been quite a significant number of patients that have been treated with CAR T cell therapies, and they don't seem to have adverse effects on the patients years out, besides the patient is somewhat immunosuppressed in different ways, but otherwise, they don't seem to cause much harm.
Thank you. And another question, what core discoveries enabled this type of therapeutic protocol to be developed versus the more, quote, traditional chemo?
Yeah, there was a lot. You know, the history of this therapy called CAR T cell therapy, as with most things, started about 30 years ago. And a gentleman named Carl Jude, who's at the University of Pennsylvania, was a pioneer behind this. And he originally developed it for HIV. But along that timeline, in the late 90s and the early 2000s, there were some really devastating gene therapy trials that they caused cancer in some patients. And very unfortunately, a young boy died at University of Pennsylvania, and it caused the whole field to take a step back. And I think that was both, good for the field to make sure that we're evaluating these things and they're going to be safe. But it led to, I think, the most persistent people in the field were able to eventually get these things through. And over the course of sticking with it for 30 years, he eventually developed it for cancer and is the inventor behind the Novartis product. And so a long-winded way of answering that question is that the core discoveries were in everything from, how do we handle genes, which is very important. How do we make sure that we're doing it safely? And it was involved in fields from HIV and virology through oncology.
Thank you. And another question, is Calibr dedicated to new pediatric oncology therapeutics?
Yeah, I would say we have different approaches to therapies which will be very relevant to diseases which are very impactful to pediatrics. For instance, this CAR T cell therapy, we are also looking at in a different type of blood cancer called AML. And AML is very devastating in pediatric patients. And so we are looking at therapies for children.
Thank you. That's all the questions I have at this point. Thank you, Travis. That was a most interesting presentation, and I also thank the audience for the wonderful questions.
Yeah, thanks so much for the opportunity to come back and talk about it. And thank you again for the support from ARCS.
And thank you, Dr. Travis. I've seen you on occasion at some of our virtual field trips. So it's always nice to see our Scholar alum tune in when possible. So really appreciate that.
Absolutely.
Is that all right if I ask a question, Holly and Ingrid?
Sure.
Dr. Young, I was wondering if you had heard of the Livingston Wheeler Clinic that deals with cancer, and they have made vaccines. This is in the 50s and 60s and 70s, using a patient's own cells. They've developed a vaccine to help fight the cancer. And so it's similar to what you're doing, I think, with using a patient's immune cells and kind of reworking it a little bit to increase the immune system in a person's body by healthier practices. I don't know, are you familiar with Dr. Virginia Livingston Wheeler? Have you heard of her?
I've heard of her. I'm not directly involved in any of that type of research but I can say the world of cancer vaccines is very, very promising. And the work that's being done at Memorial Sloan Kettering, which is, I believe, where she is, and the work that's being done at UCSD in cancer vaccines, I think is at an inflection point where we're about to see some really exciting things, because like you said, it's using a lot of the same principles as what we're doing. We're using genetic engineering to kind of supercharge a patient's own immune system to recognize the tumor. And what the vaccines are doing is they're more of a re-education without genetic engineering. So they're educating the patient's immune system for how to find the tumor, both working on the same principle of using a patient's own immune system.
Thank you.
One more quick question, please. Are any of your blood cancer therapies relevant to mantle cell lymphoma?
Yes, the clinical trial that we're running right now is open to patients with mantle cell lymphoma. Yes.
What is mantle cell lymphoma?
Mantle cell is a type, it's a histology of lymphoma. And lymphoma is generally a blood cancer that grows from the lymph. The different histologies might be follicular lymphoma or diffuse large B-cell lymphoma or mantle cell lymphoma. So it's a subset of those tumors that grow from the lymph.
Mm-hmm.
Katie Madani, I think you have a question. If you unmute yourself. Well, maybe not. Oh, Tammy, you have a question. If you need to unmute.
Yes, I'm just wondering, I used to work in clinical trials and it took years and years and years. Do you see any cutback or any more rapid approval of drugs going forward through the FDA? Because of COVID and, just things, COVID was amazing that it went so quickly.
Yeah. If there's, such a devastating year for everyone this last year, but if there's any potential benefit that comes from it, it would certainly be in the way that drug discovery is viewed, not only from people developing the drugs, but from the FDA, like you're saying. There are some things that have really caused the community, the drug development community and the FDA to kind of say, look, the things we were doing before, the ways that we were progressing before, maybe we can do these in a different way that's much more efficient. So I do think that there will be some improvements in that. And the other thing is the FDA right now, we're definitely seeing a progressive FDA. They're coming up with it's not just is a drug approved or a drug not approved, but we're seeing more and more efforts for the FDA to follow up on approved drugs and make sure they're actually achieving the effectiveness as what they claimed on the label, which I think is really, really important. And that's in oncology and other indications. And if many of you probably heard in the news yesterday, there was an Alzheimer's drug approved, the first drug approved in 20 years. Good example of where the FDA had a really difficult decision to make. Not many things are available in that space. Here's one. It's got really big questions on whether or not it's effective, but they didn't give it just a blanket approval. They made the company Biogen go back, continue to study the drug. They're going to be required to come back to the FDA in a couple of years and prove that what they approved is working. And so I think we're going to see more of that from the FDA, is more of a progressive way of thinking about things. And it's not always going to be, not everything's going to fit into the same structure. They're going to come up with ways to get things to patients in a fast way.
Okay. I think we have handled all the questions from the audience. And thanks again, Travis. It's been very, very interesting, and we look forward to hearing more from you.
Great. Thanks for having me. Thanks again.
Ingrid, back to you.
Sure. Okay. And Travis, you're welcome to stay. Leonard's going to speak for about 10 minutes now. Another short Q&A, but you probably are busy, so up to you. However, you can swing it.
No, I'm looking forward to Leonard's talk.
Great. That's wonderful.
Good.
So Leonard, who really doesn't need an introduction, he is a current scholar. We are familiar with Leonard from his introductory video, which we aired at our last SOY event. So Leonard is a current scholar at Scripps Research. He earned a BA in chemistry and statistics at Amherst College in 2018. And he's now a third-year grad student working in the Dawson and Kelly labs at Scripps Research. We asked Leonard to speak today because, in fact, he has collaborated with Calibr, and we thought that would make some nice synergy between these talks. He hasn't worked directly with Travis, but he will tell you what he has done with Calibr. And he has agreed to talk about something that Tammy corrected my pronunciation on. I'm so glad you did, Tammy. He's going to tell us what autophagy is. What is it and why do we care about it? So Leonard, over to you.
Thank you, Ingrid. That was a very kind introduction. And thank you, Travis, for a very interesting talk as well. So really excited to share my research with you all, and thank you all for listening. So I've been conducting this research in collaboration with Kristen Johnson's lab at Calibr, and I'll be discussing work that's soon going to be published about a high throughput lipid droplet degradation screen that discovered novel small molecules that activate autophagy. So it's a lot of words. I'll try to break it down in the next couple slides. So first of all, what is autophagy? So autophagy is one of the major ways that our cells degrade superfluous or potentially dangerous material in them. And in macroautophagy, as you can see in this figure, the material that needs to be degraded, which for example can be toxic aggregates, protein aggregates, or damaged organelles in the cell, they're sequestered into de novo synthesized double membrane bound autophagosomes that deliver their contents to the lysosomes for digestion. And these lysosomes contain enzymes that break down the cargo back to basic nutrients or energy that can be recycled in the cell. So autophagy is crucial for the intracellular clearance of burdensome proteins in eukaryotes. So previous research has found that problems arising along the autophagy pathway, as you can see here, are linked to neurodegenerative diseases such as Parkinson's and Alzheimer's, among others. And in the figure here, you can see that there are different proteins at various stages of the autophagy pathway, denoted by these lightning bolts. And these proteins are mutated in the diseases. So we hypothesized that the efficiency of autophagy may be critical in preventing neurodegenerative diseases. So at the molecular level, many neurodegenerative diseases are caused by aggregation of proteins or abnormal accumulation of lipid droplets. And we hypothesized that we could ameliorate these molecular phenotypes by activating autophagy, because autophagy is our natural way of degrading these toxic buildups. So we aim to develop small molecules that can activate autophagy or increase the amount of autophagy that is happening in order to clear harmful cellular components that drive neurodegenerative disease progression. So a former postdoc in the Kelly lab, Rachel Botham, in collaboration with the Johnson lab at Calibr, developed a high throughput lipid droplet degradation assay to find novel hit compounds that activate autophagy. So the key technology in this assay is this fluorogenic lipid probe, which only becomes fluorescent inside a hydrophobic environment like a lipid droplet. So when it's in water, it has no fluorescence, but then in a lipid droplet, it turns green. And here's some photos, representative photos that we took. And as you can see, when we feed the cells with 10 micromolar oleic acid, where each blue circle here is the nucleus of a cell. So here, maybe we got like 10 or so cells. We see a basal level of fluorescence where each dot is presumably a lipid droplet that has been interacting with this fluorescent probe. So we see a basal level of fluorescence here that gets reduced significantly when we treat the cells with the autophagy activator. So that suggests that the autophagy activator is reducing the number of lipid droplets in these cells. So Rachel and Kristen Johnson tested about 1 million compounds in Calibr's small molecule library in this assay. This includes the reframe library that Travis was talking about in his talk. So first, these very flat pancreatic cancer cells, SW1990 cells, are plated and then fed with oleic acid. And then they're incubated for about a day so that the lipid droplets can accumulate in the cells. Then they're treated with the compound. So out of the 1 million compounds, they're treated with all of them at 4 or 5 micromolars at 1 dose for about 20 hours. And then after the 20-hour treatment, the fluorescent dye is added and the cells are prepared for imaging. And then after about an hour of preparation, they can be imaged. And this all happens in an automated way. So this is all happening via an automated system. We're very lucky that Calibr has these automated systems that allow us to treat with so many compounds and test so many compounds. So out of the 1 million compounds that they started with, they found about 700 primary hits, which means that these small molecules reduced the lipid droplets in the cells by at least 50%, and they did not kill the cells. They were not toxic to the cells. And then they did some follow-up steps, which I'm not going to go into too much detail about, but ultimately we got to 68 compounds. That's where we're at right now. So after the follow-up steps, we get 68 small molecules that both degrade lipid droplets, and we have shown that they also activate autophagy. So with these data, we think that we have discovered 68 compounds that activate autophagy, and these are new compounds that have not been reported in the literature before. So what are we doing now with these 68 compounds? So first of all, 68 is a lot of compounds to work with. So the Kelly lab is kind of triaging with the help of Calibr to first decide which ones are the most promising to potentially be developed into molecules that can go into the clinic, for example. So we're trying to learn more about them with respect to how they activate autophagy. And we're also collaborating with other labs who can test these compounds in biological models that are relevant for neurodegenerative diseases. So these two figures these two big boxes depict some of the experiments that we're performing to learn more about our hit compounds. And the titles of each one are mechanism of action and neurodegenerative disease models. I think neurodegenerative disease models is more straightforward than mechanism of action. Mechanism of action just means how the compound interacts with the cell biochemically so that it ultimately activates autophagy. So what are the steps that happen before we see autophagy activated in a biochemical sense? So with that, I just want to thank my boss, Jeff, and all my coworkers in the Kelly lab and my collaborators. And I'd like to thank you all for your attention and for funding me for the past 2 years. It's been a pleasure meeting you all. Thank you.
Wonderful. Yeah, I meant to say that Karen and Robert Bowden have funded Leonard this year.
Oh,
Karen and Robert, thank you.
Mm-hmm.
yeah.
So any questions? Travis, do you have a question? This one was a little over my head.
So Leonard, what, the thing that I love about the autophagy program, the synergies, I talked about how we were sending that library out around the world, but I didn't talk about the work that we're doing right here at home with Scripps. So I'm glad, Leonard, that you brought it back home to talk about that work you're doing with Kristen. I think the potential for this system is just, it's so wide ranging. That's what makes it really exciting. Leonard, what specific disease indications are you most enthusiastic about? You mentioned neurodegenerative disorders, but are there specific indications that you're really enthusiastic about for this platform?
I think our most promising data, which is what makes it interesting to me, is in this prion protein aggregate model. So just going through it really quickly, this is with the Encolada lab, also at Scripps, and they have a prion protein aggregate model where they can transfect neurons with this construct. Sorry, this is going to be a little bit more technical, and this form causes aggregates to form in the axons of the neuron. So you see here there are a lot more aggregates in this with cells transfected with this construct relative to the wild type prion protein. And our compound has been able to reduce the number of aggregates in a dose responsive way in these neurons. So I think that's our most promising lead right now. And that's what we're going to put in the initial publication.
And mostly Alzheimer's, you're saying?
Yeah. So I think this disease model is the familial prion protein. So I think it is related to Alzheimer's. I'm not completely sure.
Yeah, beautiful work. I think it's going to have wide-ranging impact.
Thank you.
Leonard?
Yeah.
I have a comment. One, I now know what autophagy is, but also how to say it. So I really want to thank you and Ingrid and Tammy for that great information. And I want to compliment you on a wonderful presentation. I mean, obviously you are a bright young scholar and one of tomorrow's leaders, but you had a lovely way of explaining this to us. And we're not all experts in biochemistry and stats as you are. And so I think it's really important as a young scientist that you have that ability to bring your talk down to the level of the audience. And I don't say that in any diminutive way for our audience, but it is difficult to understand and you've made it understandable. And that's a gift. So thank you. And now I have a couple of questions from the audience. One is, I now know what autophagy is and why we should care. It has tremendous potential. Is MS a potential target disease for you?
We don't have any collaborators on that at this time, but that's more that I think we're always looking for more collaborations, right? To try our compounds in now that we've discovered so many of them. We're not a biology-based lab. We're kind of more of a chemistry based lab. So we're always happy to collaborate with more labs, and hopefully some of them come to us who have those disease models.
Thank you. Will this type of therapy eventually be able to be administered in the womb based on DNA testing that would show a marker for a specific disease?
Yeah, so since these are hit compounds that came out of a screen, there's been no optimization done to test whether they are going to be safe and effective in humans. But that's the type of work that we would like to continue doing in order to get it to that point. So that's going to take quite some time, but it's in the works.
Terrific. Excellent presentation, Leonard. Where will your research be published?
We're aiming for Cell right now. That's the publication we'd like to take this to, but we'll see whether it gets accepted or not. Yeah. And I'm happy to share with you all once, you know, wherever it gets published, and once it is accepted, I'm happy to share that manuscript with you.
Very good. We will look forward to that.
Thank you.
And I, like Travis mentioned, I'm really impressed with that library that Calibr has put together. And when you see how you started with 1 million compounds or samples and did the work to get to the 68, it's pretty impressive. And it's wonderful that Calibr in really a few short years has done so much.
Yeah.
So
really,
congratulations
we're really thankful
to,
for that collaboration.
to you both for that.
Thank you. I can't take the credit for building the library. That was actually, coincidentally, that was my wife who was worked on that library. But she's always really interested in the impact that it's having, and it's great.
Okay, I don't see any more questions here. Are there any from the audience directly that aren't in the chat?
Leonard, perhaps you should stop sharing your screen, then we can see better. Yeah, there we go. Oh yes, we have questions.
Oh, okay.
Looks like Lori. Unless that's from before.
I think that was an earlier question. At least I saw it before, but are
Yeah,
there
that was earlier.
okay. Are there any other questions from the audience at this point? I don't think so.
No.
No, I don't think so. A lot of great job, Leonard and Travis. Thank you. Everybody is saying that. And we look forward to hearing more from both of you in the future. And thank you again for taking the time to share your work with us this morning. We really appreciate it.
Great. Thanks, everyone. Nice job, Leonard.
Thank you.
Thank
Very
you
much.
both.
Excellent talk, Travis. Thank you.